All right, so welcome to the Heredity Study Party. I am going to start on page 136, chapter 8, section 12. So, we first looked at mitosis. Remember that mitosis is the process of producing two identical daughter cells, either for asexual reproduction, for growth, or for repair. In contrast, meiosis is the process of producing haploid gametes for the specific purpose of sexual reproduction. And so even though we've looked at these two topics separately, it would be worth it to do a little compare-contrast in your head, because collectively, when we look at the class as a whole, we will want to look at meiosis versus mitosis. Okay? So remember that the big picture of meiosis is to produce haploid gametes. The purpose of making haploid gametes is twofold. One, to keep the chromosome number the same. Otherwise, the chromosome number is going to double with each generation. Okay? That creates its own set of unique features, and we'll get to that particularly when we start talking about plants and we start talking about polyploidy. So if we were polyploid, we would have twice as many chromosomes or 3 times as many chromosomes as the original parent. Okay? There are not many animal species that do that. Okay? So having the chromosome number keeps the chromosome complement the same from generation to generation. The other purpose of cutting the chromosome number in half is so that we have these haploid gametes that recombine by fertilization to make a diploid zygote, and evolutionarily what that does is increase genetic diversity because we're mixing half of the genetic complement then from each parent. And so sex and sexual reproduction is the basis for genetic diversity, along with mutation. Okay? So those are the reasons why we would do meiosis in the first place. All right? So some vocabulary words just in general. Your typical body cell is called a somatic cell. You've got 46 chromosomes in 23 pairs. 1 pair from mom, 1 pair from dad. The chromosomes are numbered roughly by size, 1 through 22. Pair number 23 are your sex chromosomes, X and Y. Okay? You have 2 chromosome number 1s, 2 chromosome number 2s, 2 chromosome number 3s. Those are called homologous pairs. Homologous because they have the same genetic loci on them. Okay? The gene for whatever is located in the same place on both chromosomes. However, they could be different alleles for that same gene. Right? So homologous, but not necessarily identical. Right? And in the picture at the top of page 137, figure 8.2, they're showing you a homologous pair that is already duplicated in the S phase. And so you can see sister chromatids held together by the centromere, and the different colors are showing you not different genes, but different regions of the chromosome. Okay? Now, the locus, remember, is, or plural loci. The locus is the location of a gene on a chromosome. Okay? That's not going to change between homologous pairs. Okay? Now, chromosome pairs 1 through 22 are called autosomes. The X and Y chromosome in pair 23 are considered the sex chromosomes. Section 813 gives you some additional vocabulary words. Okay? A diploid cell being 2n. So what is n for a human? 23. Your somatic cells are all 2n, which means they have how many chromosomes? 46. Okay? So you are diploid, you are 2n. Okay? A gamete is either an egg or a sperm, and it would be haploid, 1n, in humans, 23. Okay? When two haploid gametes fuse, that's the moment of fertilization. That single fertilized cell is called a zygote. We'll talk about development farther down the road in the spring after Christmas, but so the zygote is just the single fertilized cell. That cell is instantly going to divide by mitosis to produce identical daughter cells. As soon as that zygote starts dividing, we call it an embryo. Now, definitions vary depending upon what realm of biology you're in, but once the embryo begins to develop organs and organ systems, then we call it a fetus. And we call it a fetus until it's born. Okay? And so that's just a little bit of distinction there. Notice the life cycle diagram. And I want to point this out. It's figure 8.13. I want to point this out because as we look at life cycles of organisms throughout the year, you'll see that they use the same pattern all the book long. Diploid is always shown in blue, and haploid is always shown in that tan. Okay? That's not worthy of memorization for a test, but it is helpful to know that that's the structure of this textbook to help you study. So you can see that we go between diploid and haploid either by meiosis or by fertilization. Okay? So those are the processes by which we cross back and forth. If you look at page 814, or sorry, not page 814, section 814, page 138 and 139, you see a really nice diagram with descriptions of the process of meiosis. So I just want to point out a couple of the highlights. Highlight number 1, meiosis happens in 2 parts, cleverly named meiosis 1 and meiosis 2. Okay? The purpose of meiosis 1 is to separate the homologous pair and reduce the chromosome number. She said pausing because everybody's writing madly.
Wait,
so let me say it one more. Let me say it one more time. Okay, there's 2 steps to meiosis: meiosis 1, meiosis 2. The whole point of meiosis 1 is to separate the homologous pairs, which reduces the chromosome number. So meiosis 1 is how we go from diploid to haploid. The whole point of meiosis 2 is to separate the sister chromatids and so meiosis 2 is identical to mitosis. Shall I say that one more time? The purpose of meiosis 2 is to separate the sister chromatids, which makes meiosis 2 the same as mitosis. Okay? Yes?
So meiosis 1 goes from diploid to haploid.
Mm-hmm.
So then wouldn't, instead of having half of the amount of chromosomes, wouldn't at the end of meiosis 2 the cells have a 4th amount of the chromosomes?
No, because the chromosomes, so one chromosome after S phase and interphase is still one chromosome made out of two sister chromatids.
Oh, okay.
Ah, it's the C words again. Does that make sense?
Yeah.
So, even after, so we're going through, so the cell's going through interphase. G1, growth. The cell's doing what the cell's doing, busy making proteins. The DNA is all unwound, so we can access the sequence of A's, C's, T's, and G's. We can get at the directions to build proteins. Then we go through S phase, where the DNA replicates. We still have the same number of chromosomes, but because the DNA has replicated, we have two sister chromatids held together by a centromere.
So there's 46 chromosomes
Right.
that each are made up of two sisters.
Yes, so after S, there are still 46 chromosomes that are made out of two sister chromatids held together by a centromere. The cell goes through G2, where it's still doing what it wants to do. It's preparing for mitosis. Okay? But in this case, we're not going to go through mitosis, we're going to go through meiosis.
How did they choose to do that?
Ah, well, how does a cell decide to do meiosis or mitosis? It depends on what kind of cell it is. If it is an autosomatic cell, a body cell, it's only going to do mitosis. The only cells, and we're going to stick with humans, the only cells in a human body that do meiosis are special cells within the ovary or the testes.
So they have the 23rd chromosome?
All cells have 46 chromosomes, 23 homologous pairs, including the sex chromosomes X and Y. Okay? But only the cells in the testes or the ovaries will ever undergo meiosis to produce eggs or sperm.
So it's just where they are.
Right, and so, which is a really interesting developmental question, which is well beyond the scope of our book. How does a cell know? Because remember, you started out as one single cell. Every single cell in your body has the same DNA, unless it's been mutated along the way. How does a cell know that it's an autosome? Or how does a cell know that it's a cell in an ovary or a testicle and it's supposed to undergo meiosis? Isn't that a crazy cell question if you think about it? Because they're all genetically identical. How do they know this question? We'll return to that a little bit more when we get to molecular genetics, but it's a really interesting question that researchers are still working on. Okay, so couple clarifying questions, or was that your question?
No, I haven't. I heard somewhere that girls are born with all of the eggs they'll ever have.
Ah, yes, and we will get to that in terms of the timing of meiosis. And it is true that a human female at birth is born with all of the eggs that she will ever have, and they are arrested at metaphase 2. Ooh. So they have already, before birth, all the eggs in a female fetus's ovaries have already undergone meiosis and are stopped. Okay, now, how does that happen? Another interesting question, well beyond the scope of this class, and are arrested in meiosis, or in metaphase 2. Males, the cells in their testicles, don't undergo meiosis until they reach puberty. Another question.
Yeah, does that mean that that female fetuses undergo more cell activity and division than a male?
Only by virtue of the fact, remember, that even those egg cells have not undergone the full process of meiosis. Mm-hmm. They've begun the process but not finished. So just all of those egg cells, those 200,000 or however many egg cells there are, have begun that process but not finished it. Okay. All right. So other key features to point out. Interphase is interphase. regardless of the cell. We okay over here?
Yeah, I just had a realization.
What? What is your realization?
I can't imagine having, how many was it?
Oh, 250,000 children?
Isn't that what you said? No.
Oh.
Never mind.
No, it's
All right.
not
Yeah,
a thing to
that's
say
what
out loud.
All right, we'll just move politely on. All right, so I'm moving on. So interphase is the same for both. Prophase is the same for both. During prophase, notice that we're beginning to have what's called a tetrad. So a tetrad is an additional word. A tetrad is where the two homologous chromosome pairs line up. Tetrad meaning 4. So the 4 sister chromatids are lined up. And this is where crossing over can occur. In metaphase, in prophase 1, in metaphase 1. In metaphase 1, the key difference is that the homologous pairs line up on the metaphase plate. In anaphase 1, the sister chromatids stay together, but the homologous pairs separate. And so the chromosome number is halved in anaphase 1 because the homologous pairs separate. In telophase, okay, and cytokinesis, okay, we separate the two cells. They are now haploid. Notice, though, between meiosis 1 and meiosis 2, we do not go back to interphase. There's no need. The DNA was already duplicated in S before we started. And so we instantly go, hold that thought for just a second, we instantly go from telophase into prophase 2, metaphase 2, sister chromatids line up on the metaphase plate. Anaphase 2, the sister chromatids separate, the centromeres separate. And then we have telophase 2, cytokinesis again. And then the cell is a gamete. Okay? So I'm going to pause here and answer questions, and then we'll move on. So this is recording 2 of the Heredity Study Party. I'm on page 140, section 815. Okay? So in this section, we're really going back to the story that we began with. And that's the idea that mitosis and meiosis have similarities and differences. And so really this is a summary of where we began. Mitosis produces 2 identical daughter cells for either asexual reproduction, growth, or repair. We replicate the DNA, the sister chromatids separate. In meiosis, we cut the chromosome number in half, going from diploid to haploid. The purpose is to make a sex gamete that will then recombine with another sex gamete during fertilization to increase genetic diversity during sexual reproduction. And so we have meiosis 1 and meiosis 2. Remember that in meiosis 1, tetrads line up, tetrads separate, chromosome number gets halved. In meiosis 2, essentially the same process as mitosis. Okay?
Do you know the reason for crossing over?
It adds even increased genetic diversity.
They just do that because they know to do that.
Is there like a specific location that they normally break at, or like a?
So there is no specific location, and I'm going to pause that question until we get just a little farther, and let's deal with it. We'll talk about it more when we get to that section. So, section 816 on page 141 is really dealing with the issue of genetic diversity. And so what you're looking at in figure 8.16 is you're looking at various possibilities. Here they're showing you a simple cell with a chromosome number of 4, N would equal 2. Okay? And it's showing you just 2 chromosomes, the red one's from mom, the blue one's from dad. And depending upon how the tetrads line up during metaphase 1 determines what combinations of chromosomes will be in the gamete. And so you can see that there are 4 different possible combinations with 2 chromosomes. Can you imagine, mathematically, how many possibilities without crossing over there would be?
64 trillion.
Yes, 2 to the 23rd, or 64 trillion. That's even without crossing over. So what are the odds that you would produce this genetically identical gamete, even without crossing over?
Almost 1 in 1.
Slim to none, right. Yeah, the odds are so unlikely.
Has there ever been cases of that?
Well, we wouldn't necessarily know that. That would be really difficult to tell because we would have to sequence the DNA of every single person. And let's say, for example, because I'm me, let's just say I produced in my lifetime 2 genetically identical eggs. Well, I've only actually ever gotten 2 of my eggs fertilized that I know of, right? Well, no, I could have had other eggs fertilized, but they could have miscarried before I ever even knew I was pregnant, which happens to about 1/3 of all fertilization events. Okay? So that I know of, I've only ever had 2 fertilized eggs. 1 boy, 1 girl, right? I've produced a fair number of eggs in my lifetime, but I would never know what their genetic component is, right? So it would be virtually impossible unless you harvested those eggs, destroyed those eggs, sequenced those eggs to know if they were the same. So all we're left with really are mathematical probabilities. Slim to none. Actually, probably none to none. Yes, Emma?
Wouldn't the father have to have 2 of the exact same ones as well?
Well, right. To produce 2 identical twins separated, right? Okay. So both the mother and the father. So that would be 64 trillion times 64 trillion.
So this is just like not a good chance.
Plus there's crossing over.
It happens.
And there's always crossing over. Okay? Yes.
So with identical twins, that's the one egg gets fertilized and it breaks.
And then it separates. Correct.
With fraternal twins, do 2 eggs get fertilized?
2 eggs get fertilized. So with maternal twins, identical twins, 1 egg gets fertilized by 1 sperm. In that early embryonic process, remember cells like to stick together. Sometimes if those cells get separated in the early cell stages, okay, then you can produce 2 identical individuals. Those would be identical twins. Okay? Fraternal twins, 2 eggs released by the woman get fertilized by 2 different sperm. They could be both female, they could be both male. You could have 1 male, 1 female. Okay? But they're not going to be genetically identical. They just happen to be in the same womb at the same time.
And that's because 2 eggs got released.
2 eggs, right. Just by chance. Yes?
Identical twins have to be the same sex, right?
Yes, because they're genetically identical.
Yeah.
Right? So that's not an option. Mm-hmm.
So, like a brother and sister who are twins, they're fraternal then?
They are automatically, yes, you can know automatically that they are fraternal. 2 eggs, 2 sperm.
Is it possible to have identical triplets or more than just identical twins?
Generally, I mean, is it possible? Yes. Not very likely. Now, it has been known to happen to have triplets where 2 of them were identical and the third was fraternal.
So then there were 2 eggs.
So there were 2 eggs fertilized by 2 sperm. One of the embryos split and became identical.
So what about John and Kate plus 8? Are they all?
That was
Okay, so John.
Oh, that was in a petri dish? Yeah.
Yes, so here's what happens. When you get multiple births, octomom or whatever, what you're getting, what's happening is those eggs have been harvested medically, surgically removed from the ovary of the woman. They were induced chemically, okay, so that they would finish, the follicles would be produced. They harvest the eggs, they put them in a petri dish, and then they get sperm, fertilize all of those eggs, get embryos, make sure those embryos are viable, and then they collect them and implant them. Okay, so this would be in vitro fertilization. And if that happens, if someone goes through that process, they generally implant more than just one, just to be safe that one of them. So it's not uncommon to have multiple births. Okay. On my wiki, on the heredity and on the genetics and evolution page, there is a video, a bonus video, like you guys have time, but it's there, and it walks you through an in vitro lab, and it talks about the security to make sure that the right mom gets the right eggs, and mix-ups don't happen. Yeah, yeah, and
one of those awkward situations with blood typing.
right, and
babies daily given to the wrong parent.
so right, and so it talks about the security measures. So if you're interested in the process of in vitro fertilization, I encourage you to go to the wiki and watch. It's not super long; it's maybe 12 minutes or so, and watch that video. It's a very interesting process, and it'll show you how they actually fertilize the egg and fertilize the sperm. Or not fertilize, well, fertilize the egg with the sperm, and then they show you how it develops, and it walks you through that whole process. Okay? So, back to the book on page 142, section 817. It's back to the idea of genes on the same loci. And it shows you how you could have different alleles on the same loci. So I'm looking at figure 817A. And so we can see that different chromosomes could have different alleles for the same gene. Okay? And in section 818, excuse me, it's talking about crossing over. So if there were already 64 trillion possible combinations without crossing over, you can see that what happens is we have little X-shaped regions, and those are called chiasma. Plural is chiasmata. Okay, so a chiasma is the spot of crossing over. And if you look at figure 818A, you'll notice that there are multiple crossing over events happening, which is not uncommon. Okay, and in figure 818B, they're showing you the process of crossing over. Notice that on this particular chromosome, genes C and E are fairly far apart. And so crossing over could occur in lots of different places between location C and location E. If they were super close together, the number of different possible places for crossing over is much less. So the probability of crossing over goes down. Yes?
So crossing over can occur anywhere between, either at or between the different loci?
Mm-hmm. But remember, there's probably hundreds of thousands of loci between C and E in that diagram.
Yeah.
Because those aren't the only 2 genes on that chromosome.
No, but there could be crossing over anywhere in between those 2.
Anywhere along that chromosome, yes. And so between C and E, you could imagine they're showing you one crossing over location, but could you imagine a crossing over location closer to C or closer to E? And so there's many more possible places the farther apart loci are.
Okay.
Yes?
So for a specific gene, there's only 2 alleles, but there's a lot of genes.
No, not necessarily. For any gene, there's often 2, but there could be multiple alleles, just like the ABO blood groups, right?
Yeah.
So multiple alleles is a possibility.
But that would go on one loci.
That would still be on one loci.
And then could
Yeah, so the allele for ABO blood group, IA, IB, or little I, are all going to be on the same location on the same chromosome.
okay.
I forget which one, but
And then could a crossing over occur on a loci, or can it only occur between?
it generally only occurs between. Because we wouldn't want
if
to...
that happened, would the gene disappear?
Well, so, right, and usually what would happen would be some kind of mutation where the chromosome would actually be broken. Okay, so far so good? So, a karyotype, section 819, basically it's an inventory of the individual's chromosomes. And so we capture a lot of cells, but not all cells are going to be in mitosis or meiosis at that point. So we would capture, this would be a fertilized egg. So this would be an embryo. Okay. These cells would be undergoing mitosis. Still way too early to have ovaries. So nothing's undergoing meiosis. So we've got mitosis. In metaphase and prophase, the chromosomes are condensed. We can see them under a microscope. In metaphase. Okay. We can take a picture of them with a digital microscope. We can put it as a photographic file. We can move those individual chromosomes around on a computer. We used to have to actually take a real photograph, and some poor lab tech would actually cut with with scissors out the chromosomes and then tape them onto a new photograph. That's how it used to be done. I have paper and pencil activities in my files where you would do that very same thing. You would cut them out and line them up.
We did little stickies last.
Okay. Yes, if you were in my class, I forgot that I did that with you last year. If you were in my class last year, you got the little chromosome cling guys, and you got to sort them out. That was a karyotype.
If this was to happen in, like, if you could capture this at telophase or something, all of these pairs would only have a single chromosome.
If it had happened after anaphase, they would only have a single sister chromatid.
Is that right? But it's not possible for us to capture that?
You could. It's not very useful.
Yeah.
Because usually after anaphase, we're going right into telophase and they're starting to unwind.
Yeah.
And so photographically, it's very difficult to separate and sort them. And think about this. In a cell, they're all flopping sort of over each other. They don't line up as beautifully as it shows.
Yeah.
And so you can even see in figure H20A, they're not all perfectly lined up. Some of them are a little bent over and crooked. Okay, but what we're looking for then is chromosome number. We're looking for a monoploidy or a triploidy. Okay, or not ploidy, somy. Monosomy or trisomy. Sorry, not ploidy. And so a traditional trisomy would be like Down syndrome. Okay. We can also find like where if a bit of a chromosome has been broken off. Okay. And so, yes, one question.
Do they like give these out when, because like I think I have one of these like karyotypes.
For you?
Yeah.
Yep, you could, and your mom might have a picture of it. That'd be really cool.
How do you get one?
Well, so how do you get one? So the pregnant woman either undergoes chorionic villi sampling or amniocentesis, in which case what they have to do is collect those fetal cells, and because fetal cells are busy dividing because the fetus is busy growing, or the embryo is busy growing, okay, there's a good chance of catching some of those cells. Do you remember how you fixed the onion root cells, you froze them in that moment of time, and then you stained them and looked at them under a microscope, you would do a very similar process with the embryonic cells. You'd have to find some cells that were in metaphase. You would stain them and then take a picture of them, take a digital microscope picture, and then you would line up the chromosomes according to size. So they do that if they are concerned about a possible chromosomal defect, often based on the age of the parents, or based on some family history, or based on some sort of reasonable medical concern. If I just really wanted to know if I was having a boy or a girl, the risk is probably not worth finding out.
Or they could do an ultrasound.
Or they can wait and do an ultrasound, which is not always correct. But, you know, there's other ways to find out. Okay? And so they may go back and do that. You can also karyotype after birth. So, for example, some of you may still know Jamie Kaminsky, who has all kinds of developmental delays, was a student at our high school, but now lives and works with Horizons. The ends of one of her chromosomes was broken off and deleted, causing all kinds of developmental delays. And it wasn't until after she was born and wasn't reaching any of the normal infant milestones, rolling over, sitting up, responding, that they actually took some of her cheek cells and some of her blood cells and did a karyotype. We could do a karyotype on any of you. All we need is your cells in metaphase. And you could do a karyotype.
Take a certain type of cell that is more frequently dividing.
Yeah, you'd want to get one that's dividing, and so cheek cells are a nice sample because they're continually dividing. All right? So, how do we end up with not enough or too many chromosomes, a monosomy or a trisomy? On page H21, it's called a non-disjunction event. And if you look at these two diagrams, you can see that the non-disjunction event can happen either in meiosis 1 or in meiosis 2. Okay? Normally, monosomies and trisomies often impact survival, and the embryo doesn't develop properly, and so a miscarriage happens long before the embryo is born. If, however, the non-disjunction event happens with a sex chromosome, it usually doesn't affect survival much at all. And so you can see in the chart on page 147, we can have XXY or XYY or XXX or XO, meaning a monosomy. That individual only has one X chromosome. Okay?
So what happens if they only have one X chromosome?
They have something called Turner syndrome.
Oh.
All right? Normal intelligence, they tend to be shorter. They don't, they still go through puberty, but generally breast development is smaller, and often the eggs that they produce are infertile. So often a woman with Turner syndrome is unable to have children.
Mm. And,
okay.
What happens when there's like a hermaphrodite?
I'm going to answer the hermaphrodite question when we get to development. Later on. We need to know a little bit more molecular genetics. So I'm going to stall the answer to that question right now. Did you have a question, Kel?
So you said that a woman would still produce eggs, but she would be infertile?
Mm-hmm. Because so she would still produce and she would still go through a regular cycle and have periods, but the eggs themselves, even if a sperm fertilized them, they're not going to divide properly because they don't have the right number of chromosomes to line up in tetrads. And so the process just stops. It sort of grinds to a halt.
Okay, so even if they were fertilized, they wouldn't undergo meiosis.
Right. Mm-hmm. And so then in section H23, we talked a little earlier, sorry, hang on for 1 second, about polyploidy, okay, or even tetraploidy, okay. We don't really understand exactly why it occurs, except that it does, and it often occurs in plants. Most of the produce that you eat, most of the fruit that you eat are polyploid species. Bananas are triploid. How bizarre is that? Strawberries are very polyploid. Oranges are polyploid. No, but lots of fruits are, because it generally produces larger fruits. Okay? I am going to then wrap up this chapter by looking at chromosomal mutations, section H24. And if we change, now this is a change on the chromosome. Later on, we'll talk about mutations that happen at the level of the gene. Chromosomal mutations affect lots of genes. So we could delete part of a chromosome. We would lose a lot of genes. We could duplicate part of a chromosome, and we would have duplicate genes. We could invert, so we could have an inversion event. And you can see in the diagram, the chromosome, a chunk of the chromosome broke, but it got reattached in a different direction. We can have translocation. Notice in translocation, we have a non-homologous chromosome. So a bit, for example, of chromosome 9 breaks off and chromosome 22 break off. And you can see that now we have this weird chromosome, which is called a Philadelphia chromosome. Okay? That is often associated with cancer, some types of leukemia. In this particular, it's called the Philadelphia chromosome because then we have this activated cancer-causing gene.
Would it have to happen to the other pair?
Not necessarily. It could happen to just one pair of the homologous.
So, would it have to happen to the other chromosome in the pair?
No. So chromosome 9 from mom and chromosome 22 from dad ended up having a reciprocal translocation, but the other chromosome 9 and the other chromosome 22 are just fine. That did not happen. Okay?
So you could happen to get the 9 and 22 that don't have
Right. So if you
or both of them are one.
right, so it depends upon where that mutation occurred as to whether or not you might inherit from your parent. So I'm going to stop that recording, and that's the end of chapter 8. All right. So this is recording number 3 of the Heredity Study Party. I am beginning on page 154, section 9.1. We have been doing genetics long before we knew what we were doing. There was this idea of pangenesis. Okay? And pangenesis was actually back from the Greeks. You had all of these little particles called pangenes, and they traveled from every part of your body to make an egg or to make a sperm. So particles from your eyeballs would travel to the egg, your fingers, all these different parts, right? And they would make up an egg or sperm. In fact, Leeuwenhoek, when he first looked at sperm under a microscope, thought he saw a little tiny person all coiled up in the head of a sperm. Right? The little man that wasn't. All right? Pan, yeah, so from the same word pan. Okay? All right. So, experimental genetics, the beginning of genetics really begins with Mendel, who was a monk, Austrian monk. He couldn't pass his teacher test, so he became a monk. And he had lots of help. And again, we're sort of looking at some of this basic genetics vocabulary. Okay? A heritable feature is called a character, like flower color. So we have flower color, which would be the character. The different kinds of flower color, purple, white, pink, whatever, would be called the trait. Okay?
Wait. I'm confused. What's the difference between a character and a trait?
So a character is an inherited feature. So for example, flower color, height, shape, seed shape might be a character. If height is a character, tall and short would be the traits of that character.
Oh, okay. I see what you're saying.
Does that make sense? Okay. So the parent generation is called the P generation. The children are the F1s, the grandchildren are the F2. Now, I am the F1 of my mom and dad. I am the P of my children. So any given individual, depending upon what you're talking about, could be more than one thing. Make sense? Yeah? Okay. So, Mendel's laws. Okay. Some other words. Alleles, different forms of the same gene. They would be located on the same locus. You can be heterozygous or homozygous. You can be dominant, you can be recessive. Okay? A monohybrid cross would be looking at the results of one single gene. A dihybrid cross would be looking at the results of looking at two different genes. And of course, we could talk about trihybrid crosses, etc. Right? So we're looking at different crosses. We do that by creating Punnett squares and looking at probability. Okay? Phenotype, an organism's appearance. Genotype, what combination of alleles it has. So this is all basic sort of genetics vocabulary. The law of segregation says that meiosis happens. Even though Mendel didn't really understand meiosis, the law of segregation says that each gamete, each egg or sperm, carries only one copy of each allele. Right? Meiosis, because we have to cut the chromosome number in half. So each egg or sperm only gets one copy. So it's only going to have one allele.
It only carries them?
Huh?
I feel like they
But does right, but does that make sense? So yeah, the short answer is meiosis happens. Okay? In section 9.4, again, we're looking at the same vocabulary in pictorial form. So we're talking about genotype, we're talking about phenotype, it shows you homozygous dominant, homozygous recessive, and it shows you heterozygous, and it shows you the different loci for genes on the chromosome. Okay? In 9.5, independent assortment happens, and it says that alleles, different genes, will segregate separately. Now, there's a really big assumption there. They separate independently if they are on different chromosomes. If they do not separate independently, one can assume that they are on the same chromosome, that they are linked. Okay? Yes?
Is that why some traits will go together?
Right, and so some traits don't follow the normal patterns. So in a dihybrid cross, notice that we get a 9:3 to 3:1 ratio. Do you see that in the actual results down there with the P's?
Yeah.
9:3 to 3:1 is the typical phenotypic ratio if the genes don't segregate. But if you got a 3:1 ratio, you can assume that the genes end up on the same chromosome, and they end up linked. Yes?
Oh, sorry.
Oh, no, okay.
That's not.
Yes, oh, it is possible. You only have, think about, there's 100 different genes on chromosome, or 300,000 genes on chromosome 1 alone.
So wait, so you're saying that that's like genes that correspond with each other?
Genes that are located on the same chromosome tend to be inherited together, unless crossing over occurs.
And that's
just
So, independent assortment means that the genes don't segregate completely independently of each other, which is what happens if the genes are located on separate chromosomes. So in this picture on page 158, the gene for seed color, yellow or green, is on a different chromosome than the gene for seed shape, smooth or wrinkled, because they segregate to eggs and sperm. They sort out completely independently of each other.
So that's only if they're on a different chromosome.
Right. But if this 9:3 to 3:1 ratio isn't followed, you might assume that perhaps they're linked on the same chromosome and they're segregating together.
This one. Blonde hair and blue eyes for an example.
Okay, that's a really, no, that's a horrible example, because, jumping ahead, those are, eye color and hair color are polygenic as the result of many, many genes contributing to a single trait.
So would ethnicity,
Skin color?
like skin color versus
Skin-
eye shape?
Skin color is also polygenic.
Oh.
What about?
I don't understand. Can you give like an
Yeah,
example?
Okay,
Don't
So-
correspond together.
so what we have to do is we have to back up, and we should not use humans, first of all, because there are very few human traits that are controlled by a single gene. Okay, so here is one trait. So the ability to roll your tongue is controlled by a single gene at a single locus.
Okay.
You can either be a tongue roller or a non-tongue roller.
Oh, and mid-digit hair.
Okay, right,
I
or
remember that from math.
Yeah.
right, or hitchhiker's thumb. You have to be
Yeah.
careful because in math they don't always use accurate examples, I've discovered.
Oh, really?
So hitchhiker's thumb is a single gene trait, or widow's peak, or attached earlobes. And that's about it, guys. There's not that many single gene traits. And so, having a widow's peak or not having a widow's peak, your chances of having that have nothing to do with whether or not you can roll your tongue, because they're located on completely different chromosomes.
Okay.
So there aren't any. Are there any examples of this in humans?
No.
But I guess I don't know.
So let's stop. I'll show you. There's a picture when they talk about linked genes down the road.
Okay.
Okay? So let's stall for just a moment.
This dependent assortment,
Yes.
you said that can not have.
That doesn't relate to humans.
So we need to, so we need to sort of let the human examples go.
But this dependent assortment could exist.
Independent assortment happens.
But the dependent, could that exist in other species?
Yes.
If they're on the same chromosome?
If the genes are on the same chromosome, you will not get independent assortment happening, because the genes travel together on that chromosome into that egg unless crossing over occurs. Okay? And then they talk about a test cross. We already talked about test crosses with blood type from last class, determining a test cross. And so you just have to use the recessive type. Okay? In section 9.7, they're talking about the rules of probability, which you should remember from your math class. Ands and ors. Ands means multiply. Ors mean add. Okay? And so they give you an example. If you're having trouble with these rules of probability, I gave you a POGL on statistics of genetics, and it worked you through some of those problems. And so that's not something I would necessarily expect you to complete, but I gave it to you just as a support in case you're having issues with calculating. It was one of the little worksheet handouts.
Oh.
Okay? It says Pogel at the top of it, but don't worry about that. Okay? We can track pedigrees. We can track traits through pedigrees. We worked out Danny's pedigree at length. Okay? And so I'm not going to spend a lot of time in our review session other than to remind you that circles are females, squares are males. Vertical lines indicate offspring. Horizontal lines that connect indicate union. We'll be politically correct and say marriage. Right? If you draw an X through one, this is the only thing I don't think we talked about. Sometimes in pedigrees, if they draw an X through one, it means that they're deceased. Okay? Sometimes there's carriers. Sometimes they show carriers by coloring in half of the symbol, but sometimes they don't. All right? Some inherited disorders are controlled by single genes. Often they are recessive, which means that you can be a carrier.
So a pedigree would only express one character?
So generally, yes, a pedigree will only track one character of interest. Yes. And so if you look at page 163 at the chart, do you need to memorize these disorders? No, but you should know that most genetic disorders that are genetic, caused by a single gene, most of them are recessive. There are only a few, like Huntington's disease would be probably the one that you should remember that's dominant. If it's a dominant disorder, can you be a carrier? No. You either have it or you don't, right? Because you only need one copy of the allele to have it. All right? And so now back, and you can see how these chapters overlap. On page 164 in section 910, we're back looking at amniocentesis versus chorionic villi sampling, and they're showing you an image of karyotyping again. We can do fetal imaging and newborn screenings by looking for markers. Okay, so I'm going to stop this recording here.
Are we going to go into more detail?
Oh.
So, continuing on, I don't even remember what recording number this is. Okay, so this is memo recording 4. I'm going to begin with section 911. Mendel's laws work for the most part, but there are many, many, many traits that don't actually follow Mendel's laws. And so I'm going to list them if you have specific questions. So this part, again, is mostly review from 10th grade biology. The first is called incomplete dominance, where the heterozygote shows a trait that is intermediate or in between. The example they give you is a red flower crossed with a white flower. The heterozygote has a pink flower. Okay? The other example they give you is hypercholesterolemia, where the normal condition, okay, you can make LDL receptors. If you're heterozygous, you get a few receptors, and if you're homozygous, you can't make the receptors at all, and you have a severe symptoms of that disease. Okay, so that's incomplete dominance. We can have multiple alleles. The example are the ABO blood groups. And remember, with multiple alleles, we can also, in this case, have codominant, where what?
All the little diagrams of the chromosomes.
With the chromosomes?
And it has the little loci.
And it shows you the loci?
Yeah.
So I don't even know I forget what chromosome the blood types are on, but let's just say it's chromosome 3. I'm totally making this up. So let's say the loci is right here. Well, you get one allele at that loci. So it's either IA or IB or little I. And so just like my daughter, she has 2 chromosomes, one from me, one from her dad. On her loci, one of her loci has an IA. Thanks, Mom. and one of her loci has a little I. Thanks, Dad.
So
Right? You still only get 2 copies of that allele, of that gene.
multiple allele.
There's just
Right.
more.
There's more possibilities of what could be at that loci. Okay? And so if it's codominant, we write them as superscripts.
Can you have multiple alleles that are not codominant?
I suppose you could have,
Not
no,
really?
not really, because you wouldn't be able to tell. If we had 2 alleles, if you had co-recessive alleles, you wouldn't know,
Right. So that's
right?
like type of blood.
Yeah. Okay. We can have a single gene affecting many traits. In this example, they're giving you the example of sickle cell anemia. The word for that is pleiotropy. So in this case, if you have sickle cell anemia, notice that all kinds of stuff can happen to you. So in that case, one gene is resulting in many, many, many phenotypic characters. On the flip side, so the opposite in a way of pleiotropy is polygenic. One trait controlled by many genes. Can you see the difference? One pleiotropy is one gene, many traits, where polygenic, one gene, or sorry, many genes, one trait.
And that's why it's hard, like in humans, to define where it came from.
Most human traits are polygenic, which is why we get a bell curve kind of distribution. So height, skin color, eye color is actually controlled by, I forget, like 13 or 18 different genes, which is why we can get different shades of blues and browns, and that is why 2 brown-eyed people can sometimes have, or 2 blue-eyed people can have a brown-eyed child. Okay, it's possible. All right, in this case, they're giving you skin color, right? And it says the environment can also affect many characters. This is not epigenetics. Section 915, this is not epigenetics. This is the environment affecting gene expression. So, for example, I got a certain combination of genes for height. But had I grown up in sub-Saharan Africa and starved most of my life, I might be significantly shorter than I am now because I didn't get enough nutrition. Does that make sense?
How is it not epigenetics if it's not like...
Because so that affected the expression. So my phenotype is genes plus environment. In epigenetics, the environment is actually affecting my genes that I pass on. So just because I had tall genes, but I was raised in a horrible environment so that I never grew to my full height potential, I can still pass on, I'll still pass on those tall genes to my children.
Right.
Yeah. Okay. Does that make sense?
That's epigenetic?
No, that is not
So,
epigenetics.
That's
That's just the
chemical
environment.
tag, like the tags.
Right.
Like, you know of cases where kids are adopted from Africa and then they raised in the United States and then they go back and, like, I just know of cases.
Right. And so
they
the,
go back and they're extremely taller. And
they're much
bigger.
taller, they're beefier, they're more filled out, they're healthier. That is not epigenetics.
That's environment.
That's environment. So in this case, we're talking about your phenotype. What you look like is a direct result of your genes plus your environment. Okay? I could have much darker skin if I spent a lot more time out in the sun. But did I change my genes? No. Okay? And so in this next section, 9:16, they're basically mashing together what we know about genetics with what we know about meiosis. And you can see that this diagram is still the same. They're showing you how the chromosomes sort out during meiosis to make alleles and how we end up through the Punnett square to get our 9:3 to 3:1 ratio again. Does that make sense? So in a Punnett square, when we're writing alleles, we could draw the whole chromosome and put the allele on its proper loci. That's all they've done in this section. Does that make sense? So they're making overt the link between chromosomes and meiosis and the genes that you get. All right? In section 9:17, now we're back to our illustration. Remember I stalled us a little bit about independent assortment? And so you can see in this case, genes that are on the same chromosome will be linked together. And so notice what happens. Long pollen versus short pollen and flower color. Red versus purple. We would expect, because purple is dominant over red and long pollen is dominant over round, we would expect the inheritance pattern to be 9:3 to 3:1 because it's a dihybrid cross. And so the numbers we would expect, you can see there at the top of the chart, we would expect 2, 15, 71, 71, and 24. But those are not the numbers that we actually observed. And so we conclude, oh, something else must be happening. And so in this case, we have to have a different explanation. And so we explain that they must be linked. If they're linked, notice the Punnett square. Notice that we should get a 3:1 ratio of purple long to red round. Do you see that? So where did the other oddballs come from?
Another
Crossing over. Oh, and that's why they're present in such low numbers, because crossing over doesn't always happen. Is this making sense to you right now?
So on this, I don't understand where they got 3:1 by looking at this diagram.
Okay, when you look, they're linked together, right? So purple and long are on the purple chromosome. Red and round, both recessive, are on the red chromosome. So when we do meiosis, we can only get two kinds of gametes. The purple long or the red short. Does that make sense? And so we set up a Punnett square. Purple long times purple long will give you purple long. A purple long times a red short, or red round, will be heterozygous, but the genotype will still be purple long because of the dominant genes. Are you seeing the 3:1 now?
Yeah, I was counting like each individual purple.
Oh no.
I was like, that's
Because if I'm big A, little a,
You're still.
I'm heterozygous, but I still show the dominant phenotype, right? And so now we're looking phenotype, because remember, we can only observe the phenotype. We can't observe the genotype. And so that's where they're getting the 3:1 phenotypically. Make sense?
yeah.
And so we would only expect to see two phenotypes. The other two phenotypes show up if there's crossing over between those two linked genes. And so they're going back to showing you how crossing over happens, and so they're showing you a chiasma at the top of page 173. Do you see that? Okay. And then they're showing you an actual experiment with fruit flies. Don't worry about the details of the experiment, but notice that they're finding out how often, what % of the time are they actually getting crossing over. So in this experiment, notice that they got crossing over 17% of the time. So cleverly, geneticists say, we're going to call map units. So if we were to map a chromosome, we could map how far apart alleles are on the chromosome, and we do it, so we use map units. One map unit is 1%. So if crossing over happens 17% of the time.
Wait, where do you get 17%?
Oh, there it is.
Just in this particular example.
Okay, yeah.
So in this particular example, crossing over happens 17% of the time between these two genes. So if we were mapping these genes, we would say that they are 17 map units apart. Okay? So I am going to give you 3 genes, just as a little, I'm going to give you a little sample problem. I am going to give you 3 genes. Gene A, gene B, and gene C. Okay? And I'm going to tell you the percentage crossing over, and I want you to tell me the order of the genes on a chromosome. So, A and B have a crossing over frequency of 25%. So how many map units are between A and B? 25. Okay? The frequency of crossing over between A and C is 8%. So what's the different distance between A and C? 8 map units. The distance, the crossing over frequency between B and C is 17%. So, what would be one possible order of chromosomes on this, of genes on this chromosome? A, C, B. Do you see that?
Yeah.
Does that make sense to you? Or BCA. Right, either way, depending upon which way the chromosome goes. Correct. But C has to be in the middle based on that data. Make sense? Yeah. Super simple. All right. Oh.
So what would a question look like, though?
Well, turn the page.
Yeah, good.
Turn the page. And look, I got all ahead of myself. If you would look at section 919, they give you a sample problem. And in this sample problem, they used genes G, C, and L. And notice they gave you different gene recombination frequencies. Okay, so they would tell you the percentage, just like I did, they would tell you the percentage crossing over, and they would ask you to try to map out those genes in a way that makes sense. So you just have to make the numbers add up correctly.
Oh, so if it said map out the genes, you'd say GCI.
Right.
Because the question would have only given you the percentages.
Uh-huh.
Oh.
And you would have to decide how close those genes are to each other. Yeah, not that hard. I mean, it's super simple, really.
And then when you throw in all the vocab, though.
I know, right. Right, but you have to, but so this is why we're doing multiple choice, so you can slow down and sort out the vocabulary. Okay. I'm going to stop this, and we're going to do one last recording. So, this is recording 5 of the Heredity Study Party. I'm going to start at the bottom of page 174 with section 920. Okay? And we're looking at sex determination. We tend to think of the XY system because that's what we are. XX, female. XY, male. 50% probability. But that's not the only possibility out there. You do not necessarily need to memorize these systems, but you just need to know that there are other systems that exist. So, for example, there is an exo system in grasshoppers. If you look at that picture, females have 22 chromosomes plus their sex chromosome, XX. If a male if he has all his chromosomes, 22 plus just one X, he's male. So depending upon whether you get 1 or 2 sex chromosomes determines whether you're male or female. In roosters, the female is the heterozygote and the male is the homozygote. So we use Z and W instead of X and Y. I know, crazy? And in bees, sex is determined by chromosome number. Only the fertilized eggs become female, the queen. The unfertilized eggs are born male and they're the workers. Okay, so there's different ways to determine sex, not just the human way. But in section 921, sex-linked genes inhibit a unique pattern of inheritance, and they give you 3 examples of sex-linked problems down at the bottom of the page. And please notice that we write sex-linked. So sex-linked assumes that it is on the X chromosome. So if we were to define, explain, and give an example of sex linkage, bless you, we would say that the gene is located, a sex-linked gene is located on the X chromosome. And notice that when we solve Punnett squares, we have to write it as a superscript, either the dominant or the recessive trait over the X. Right? Sex-linked disorders affect males more than females because females have the option to be carriers because they have 2 copies of the X chromosome. Males only get 1 copy, and therefore they either have the trait or they don't. Okay? Hemophilia, red-green color blindness. Danny gave us a great example of that, but not all color blindness is sex-linked. Okay? Jack mentioned earlier tonight about the Y chromosome. So we can follow male lineages through the Y chromosome. And here's where we get back to the human example of he who leaves the most reproductive offspring wins. Or the penultimate example of raping and pillaging. Right? Because it says that, most people, 16 million men can trace their males can trace their lineage to Genghis Khan. The same thing is true of a warlord in Ireland. That was the other example they gave you. Okay? So, Y inheritance. In terms of humans, again, not many genes on the Y chromosome. Hairy ears is 1. And that takes us to the end of chapter 9. So that was a super short little last study party. What we have not covered in this study party that we will cover in class tomorrow includes, we'll do a review of epigenetics that you did the web quest on. I will talk to you about epistasis, and we will calculate mathematically how to calculate chi-squared problems. So make sure you bring your calculator, and we'll work on that. There we go. So I'm going to stop this recording.